Okay, well, welcome everyone. Thank you for joining us today. My name's Tracy Smith. I am the Collaboration, Education, and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. And in this seminar series, we highlight new papers and methods from our center that we believe to be of interest for the broader community. Today, we focus on a recent study of somatic mosaicism that uses single-cell multi-omic technologies to profile individuals with clonal hematopoiesis to identify specific epigenetic and transcriptional events that lead to biased differentiation and aberrant cellular phenotypes. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. And our guest today is Dr. Anna Nam, an assistant professor of pathology and laboratory medicine at Weill Cornell Medicine. She received her MD at the University of Missouri. During medical school, she conducted immunology research at the NIH through the Howard Hughes Medical Institute NIH Research Scholars Cloisters Program. She completed her residency training in anatomic pathology and fellowships in hematopathology and molecular genetics pathology at Weill Cornell. During her clinical training, she conducted postdoctoral research with Dr. Dan Landau at Weill Cornell and here at the New York Genome Center. And her current laboratory seeks to elucidate the mediators of genotype to phenotype relationships in hematopoietic neoplasms. And now, Anna, I'll hand it over to you.
Great. Can everyone see my screen? Perfect. All right, thank you so much for the introduction and the opportunity to share our work. This is a work that I helped co-lead as a postdoctoral fellow in Dan's lab and continue to do so as I transition to my current position as assistant professor in pathology. This is a highly collaborative effort, and so I wanted to start by acknowledging the amazing team who co-led and co-supervised this work. Neville is an MD PhD student who I worked closely with and was instrumental in various aspects of the analysis and in optimizing the analysis pipeline for the genotyping of transcriptomes that we called Iron Throne. Franco is a postdoctoral fellow in Dan's lab who worked closely with CICA to help analyze the epigenetic data modalities. We're also grateful for Robbie, who is another MD PhD student in Dan's lab, who pushed forward the experimental aspects of the project, especially after Ronan, who started the work that was started by Ronan after he transitioned to leading the single cell genomics core at MSKCC. I also want to highlight Tarek, who was a fellow in Irene Gabriel's lab, and he was instrumental in coordinating the samples, identifying the samples, and providing us with the critical clinical data for these patients. He's now at Mount Sinai, so I'm sure that we'll be hearing about more of his work in the following years. And as always, we're grateful for Dan's guidance on this project. So across most cancer types, cancer initiation and progression represents a clonal evolutionary process via acquisition of somatic driver mutations over years. In the recent years, several landmark studies have established a high prevalence of cancer driving mutations in normal tissues across organ types associated with aging. These studies demonstrated the presence of somatic mosaicism, providing a novel avenue for us to study early clonal events in human tissue with the obvious implications for the possibility of potentially preventing cancer development if we can understand how to target these early clonal outgrowths. Now, this clonal mosaicism in the blood system is referred to as clonal hematopoiesis, or CH, in which the patients exhibit no clinical or morphologic abnormalities in their blood cells. CH marks the earliest identifiable event in clonal evolution that may subsequently lead to the development of myeloid malignancies. Consistently, somatic mutations in CH overlapped with recurrent drivers of acute myeloid leukemia, or AML, with DNMT3A mutations representing the most frequently identified events in CH, thus providing a window of opportunity for us to study the impact of leukemia driver mutations at the earliest stages of neoplasia. However, this is challenged by the fact that mutated cells often represent a minority in CH and are morphologically and immunophenotypically indistinct from wild-type cells, and thus cannot be isolated for downstream analysis. While single-cell RNA sequencing is well established as a powerful tool for interrogating complex populations, such as the hematopoietic stem and progenitor pool, it is not able to distinguish the mutated cells from the wild-type. To overcome this challenge, we leveraged our multi-omic single-cell sequencing to profile the mutation status and transcriptomes for the same cells at high throughput. We call this genotyping of transcriptomes, or GOT. First, we modified the procedure by spiking in a gene-specific primer during the cDNA amplification step, particularly for lowly expressed genes. Then we siphon off a small portion of the barcoded cDNA library and amplify the locus of interest. The genotyping data and the transcriptomic data can then be intersected via the shared cell barcodes. Armed with this technology, we asked, how do CH mutations alter the hematopoietic differentiation landscape and prime these mutated cells for malignant transformation? Notably, CH specimens with enriched hematopoietic stem progenitor cells are rare, as individuals with CH have no indication for a bone marrow biopsy. To circumvent this limitation, we screened 136 stem cell grafts collected for autologous transplant for multiple myeloma patients who were in remission. This was again in collaboration with Irene Gabriel's lab. We identified 4 individuals with a common acute myeloid leukemic driver mutation, the DNMT3A R882 mutations, allowing the profiling of thousands of CD34 positive cells with GOT. However, DNMT3A is relatively lowly expressed in the 10x platform, requiring us to optimize the Iron Throne workflow to improve the accuracy of our genotyping, a work that was spearheaded by Neville. This was critical for the lowly expressed genes because these targets require a greater number of PCR cycles for us to capture the mutation locus, which means that the PCR distortions, such as PCR errors and combinations, are introduced to a greater degree than what we had observed in our previous work. You can see in this frequency pie that the expression of DNMT3A is relatively low, with most of the cells showing no expression for the DNMT3A gene here in the beige color, or 1 UMI count for the gene in the lighter orange. And the 10x procedure itself is able to capture the RA2 locus specifically in a very small percentage of this profiled cells. Whereas with the optimized GOT and Iron Throne, we're able to capture the genotyping information for a large proportion of cells that demonstrate any expression of the gene. Neville made 2 key adjustments to the original Iron Throne pipeline. First, based on the work from Ian Sudbury's group, Neville incorporated a network-based UMI collapsing algorithm to aggregate amplicon reads that likely originated from the same UMI in the original 10x library, accounting for PCR and sequencing errors within the UMI sequence. He applied this to the original species mixing experiment we performed to establish the ability of GOT to capture the mutational status in 10x in our previous manuscript. We mixed murine cells with mutant CALR transgene and human cells with wild-type CALR, whereby we demonstrated that the mutant UMIs were enriched in the murine cells, and the wild-type UMIs were enriched in the mouse cells, as we would expect. After applying the UMI collapsing algorithm, you can see that the number of cells with a mixture of mutant and wild-type UMIs, they're highlighted in the red box, were largely removed, demonstrating that this algorithm was able to remove many of the aberrant genotyping UMIs. Next, we interrogated the 10x gene expression libraries, or GEX, to identify cell barcode UMI pairs that we observed in the GOT libraries. We classified them based on whether the cell barcode UMI combinations were linked with the DNMT3A gene in the GEX, not identified in the GEX library. Or lastly, cell barcode UMI combinations that were linked with a different gene than the DNMT3A, likely representing PCR combinations in the highly amplified library. These were removed from our GOT data. And we kept the genotyping UMIs with no gene in GEX, with high supporting UMIs, taking advantage of the observation that the cell barcode UMI paired with DNMT3A have a large number of supporting reads. Based on this iron throne pipeline, then we observed that the mutant cell frequencies by GOT correlated well with the variant allele frequencies from bulk sequencing of unsorted graft products. First, we used the Surat workflow to integrate the patient samples, and we clustered these cells, these CD34 positive cells, based on the gene expression data alone, agnostic to the genotyping data, and observed the expected map of hematopoietic differentiation, including the undifferentiated HSPCs here in the dark blue, as well as the lineage committed progenitors, such as the neutrophil progenitors or the erythroid progenitors. In this highly sort of stem progenitor enriched population, we also observed these intermediate states, such as immature myeloid progenitors previously shown to be enriched for CMPs, the common myeloid progenitors as defined by flow markers, as well as intermediate states of IMPs that showed a differentiation bias toward the megakaryocytic erythroid lineage, or the ME, or the granulomonocytic lineage IMP-GMs. Once we had this map of differentiation, we projected the genotyping information as expected from the fact that DNMT3A mutations arise from the hematopoietic stem cells. Mutant cells were commingled throughout differentiation, throughout the differentiation topology. However, we did not see novel distinct mutant clusters. In other words, the DNMT3A mutations do not result in a new cellular identity, but rather modifies the existing cellular identities. Then we asked, how do DNMT3A R882 mutations alter the differentiation trajectories in clonal hematopoiesis? To address this question, we first globally assessed differentiation through comparison of pseudotime between mutant and wild-type cells, and found no significant overall differences between mutant and wild-type differentiation. Next, we asked whether DNMT3A mutations may skew differentiation at the major fork of myeloid versus lymphoid commitment. To do this, we isolated lymphomyeloid prime progenitors, or the LMPPs, and common lymphoid progenitors, or CLPs, and reclustered these cells. We found that the mutant cells were biased toward the myeloid over lymphoid differentiation, consistent with previous reports and mirroring models of DNMT3A mutations. When we looked more globally at mutant cell frequencies across the progenitor subsets, we identified that the mutant cells were highly enriched in the immature myeloid progenitors, supporting the overall myeloid bias that we observed. So we asked whether the DNMT3A mutations may result in a proliferative advantage to these IMPs, and sure enough, we observed a significant upregulation of cell cycle gene expression, specifically in the immature myeloid progenitor cluster. Further, we found that these IMPs were skewed toward the megakaryocytic erythroid differentiation, as evidenced by the transcriptional priming and corresponding enrichment of the mutated IMP-MEs versus IMP-GMs. These results were consistent with previous work led by Franco in the lab, showing that the DNMT3A knockout mirroring hematopoietic stem cells showed a similar ME bias. To explore the hypothesis that the observed ME bias, or the megakaryocytic erythroid bias, may stem from a change in transition rates into the cell state, Franco measured the transition probabilities between progenitor subtypes with RNA velocity. Consistent with this hypothesis, we identified that the transition probability of mutated IMPs to become IMP-MEs was higher compared to that of wild-type cells, whereas the transition probabilities of IMPs to IMP-GMs was diminished. Having observed these differentiation biases, then we asked, what are the transcriptional underpinnings of these differentiation skews? One of the main advantages of GOT is the ability to perform differential gene expression analysis between wild-type and mutated cells within specific progenitor subsets from the same individual, removing much of the biological and technical batch effects. Through this analysis, we identified a regulation of lineage markers such as CD9 and HSPCs previously shown to result in megakaryocytic priming in hematopoietic stem cells. We also observed enrichment of leukemia stem cell specific gene expressions demonstrating that the aberrant gene expressions observed in AML can be observed even at this early clonal stage. Gene set enrichment analysis of differential expression data within the erythroid progenitors showed an upregulation of target genes of MYC and MAX, key transcription factors for regulating stem cell self-renewal and erythroid differentiation. Next, we asked what aberrant DNA methylation changes may serve as a link between DNMT3A mutations and the observed transcriptional dysregulations. To address this, we leveraged another single-cell multi-omics platform that jointly captures the methylome, transcriptome, and somatic mutation status within the same cells. Using this technology, we identify similar progenitor subsets and genotyping information, albeit at a lower throughput. Similar to GOT, this enabled us to compare the methylation status of mutated and wild-type cells within the same patient sample. Assessment of the various genomic regions revealed that CpG islands and enhancer regions were particularly hypomethylated in DNMT3 mutated cells. Notably, Ronan had implemented double restriction enzyme digestion in order to improve the capture of the CpG poor regions, such as the enhancers, as enhancers have been previously shown to be particularly impacted by DNMT3A mutations in AML. Furthermore, we performed differential methylated region analysis between mutant and wild-type cells and promoters and identified hypomethylation of PRC2 targets. We further confirmed this by assessing the average methylation of ChIP-seq peaks for PRC2 components, SUS12 and AZH2. However, as in-code ChIP-seq tracks reflect aggregation across several cell types, we validated the preferential hypomethylation specifically impacted regions marked by the histone mark bivalency in human hematopoietic progenitors by intersecting the ChIP-seq tracks with bivalent peaks in CD34 positive cells. We then asked whether PRC2 target hypomethylation persisted through malignant transformation into AML and found that PRC2 targets were indeed significantly hypomethylated compared to promoters without PRC2 targets in R882 mutated AML. Next, we assessed whether R882 mutations display CpG sequence context specificities. In other words, are certain CpGs particularly hypomethylated due to the flanking sequences around the CpG. So Franco assessed the flanking motif of hypomethylated sites relative to the hypermethylated sites in mutated cells and identified that the CpG with T at the N+1 location was particularly hypomethylated. This result was corroborated by previously reported deep enzymology studies that identified the same T at the N+1 position as being particularly disfavored by the mutated DNMT3A. As DNMT3A R882 induced changes in DNA methylation are globally distributed across the genome, resulting in modest changes, we further hypothesized that the influence of the flanking sequence on mutated DNMT3A activity may differentially impact specific transcription factors and their binding. We therefore identified transcription factors whose binding motifs had high similarity to the hypomethylated motif and which were also highly expressed in hematopoietic progenitor cells. MYC, MAX, TFs were highlighted, as well as the HIF1 alpha aren't transcription factors critical for hematopoietic stem cell self-renewal, and USF1 and 2, which have been implicated in erythroid development. Thus, we hypothesize that the altered motif binding of these TFs may result in aberrant TF activities, as transcription factors such as MYC and HIF1 alpha have been shown to display enhanced TF binding of demethylated motifs. To address this hypothesis that TF activities may be altered by the methylation state of their binding motifs in CH, we turn to chromatin accessibility assay, which has been demonstrated to be a reliable surrogate for determining TF activities. So to validate the impact of mutated DNMT3A on TF activity, we collected Lin-negative, KIT-positive hematopoietic stem and progenitor cells from mice with and without the DNMT3A R878 H mutation, which is a murine R882 equivalent. This was in collaboration with Ross Levine's lab at MSKCC. Confirming our results in human CH, we found that the accessibility of DNMT3A R882 specific hypomethylated motif was increased in the R878H cells, including the H species, as well as specifically in the erythroid progenitors. Candidate transcription factors with high similarities scores in their binding motifs with the hypomethylated motifs, such as, again, the MIG-MAX, HIF1 alpha, ARNT, USFs, displayed enhanced accessibility in R878H cells compared to wild-type progenitors, supporting the model in which the transcription factor activities are altered based on the methylation status of their binding motifs. In support of this model, we observed a strong negative correlation between the differential accessibility of TF motifs between mutated and wild-type cells and the similarity scores of these TFs to the hypomethylated motif. Now, returning to our single cell methylation data, MYC targets were indeed hypomethylated in the mutated cells, which may underlie our finding of MYC target enrichment, gene expression enrichment in mutated cells by the initial GOT studies. And importantly, MYC motif hypomethylation corresponded with increased expression of MYC targets within the same cells, again, consistent with the previous reports indicating that MYC activity is enhanced with hypomethylation of its binding targets. One of the main advantages of these single cell multi-omics methods I discussed is, again, the capacity to compare the mutated cells to the wild-type cells from the same individuals, meaning that these cells share the same microenvironmental changes, providing us with mutant-specific information. However, since the CH samples we observed derived from individuals with previous cancer diagnosis and therapy, we wanted to confirm these results in a patient with CH without any prior cancer diagnosis or exposure to chemotherapy. It took a long time to identify a suitable sample. But we were fortunate to collaborate with Lucy Gottlieb from University of Chicago, who identified a CH sample which had a high variant allele frequency of 40% for the DNMT3A R882 mutation, meaning that 80% of the cells in the sample were affected. This meant that we could compare these CH cells to published data sets of 5 normal bone marrow CD34 positive cells. So using the sample, we confirmed that the differentially expressed genes we identified in the original CH patient samples were also differentially expressed in this CH sample compared to normal controls. In another example, we also confirmed the major finding that the MIG-MAX target genes are upregulated in the DNMT3A CH progenitor cells, especially the erythroid progenitor cells, compared to the normal control. And finally, we performed chromatin accessibility assay on these same cells and identified that the accessibility of the hypomethylated motif and the motifs of the implicated TF, such as MIG-MAX, were enhanced compared to the shuffled motif, consistent with our data in the mirroring model. So in conclusion, we demonstrate our ability to interrogate human somatic mosaicism by leveraging single cell multi-omics platforms. Through GOT, we identified differentiation skews in the mutated cells, specifically myeloid over lymphoid priming and megakaryocytic erythroid priming, associated with key transcriptional disturbances such as increased cell cycle gene expression in the immature myeloid progenitors or MYC target gene expressions in the erythroid progenitors. We also identified aberrant markers previously identified in leukemia stem cells that may enable enhanced stem cell fitness. Through our multi-omics platform that captures methylome as well as transcriptome and somatic genotyping, we identified hypomethylation of CPG islands and enhanced regions. Our analyses highlighted PRC2 and MYC target hypomethylation as potential mechanisms of activation of self-renewal and oncogenic programs that enable clonal expansions in CH. All right, with that, again, I'd like to thank everyone who were instrumental in this project that I highlighted in the beginning and throughout the talk, and I'd be happy to take any questions that you might have.
Great. Thank you so much for sharing your work with us, Anna. Yeah, we'd like to move on to our Q&A section. So if anyone has a question, please raise your hand or type it into the chat box. Just noticed my video is off. But one question that I had was about, you have a very limited sample size for the study, which is for obvious reasons, but are there plans to try to gather more samples in the future? And if you could, what kind of questions do you think we could answer having more power?
Yeah, definitely. I think there, in the field, at least in genotyping, there are a couple of approaches that people have taken. So the approach that we have taken so far is to focus in on particular driver mutation and try to have relatively robust replicates. Here, again, as you highlighted, we're somewhat limited because of the nature of the rarity of these samples. Another approach is to profile various types of somatic mutations across different genes in the context of CH, which other groups have are doing and have done, for example, in AML and Peter Galen's work. So yeah, as to your question about, are we trying to gather more samples? Yes, definitely. We have several collaborations in which we're procuring more clonal hematopoiesis samples. Again, the R882 specific mutations are particularly, these specific mutations are particularly hard to come by. So yeah, we're always looking for more, but we're also interested in understanding how other somatic drivers may impact or may result in clonal outgrowth, and whether there are sort of converging downstream effects that enable stem cell advantage.
Thank you. Another question here in the chat we have is, how does R80, I can't say it as fast as you, R882 CH affect biochemical function of DNMT3A?
Right, exactly. So I tried to allude to this a little bit. So I think I may have failed to mention that it's a methyltransferase that this enzyme is, or this gene is encoding. And so the previous studies in vitro, in which they took this enzyme, the mutated enzyme, and did sort of enzymology studies on various sort of oligos within this in vitro experiment. They identified similar findings as we did in human samples, that the flanking sequence of the CPG affects its function. So that's sort of the aspect that we highlight. But again, the other aspect is that there is, to a degree, some heterozygous loss of the wild type function of the R882. But again, the specific effect on disfavoring this N+1 flanking motif is one that was highlighted in our work.
Okay. And one more question. What marker genes did you use to differentiate mutant cells from wild type cells?
So we used the GOT method to specifically identify the mutant cells from the wild type cells. And from there, we, having been able to classify these cells based on the GOT method, we're able to compare the transcriptomes of the mutant and wild type cells based on differential gene expression analysis. And some of the markers that we highlight are CD9, for example, in the hematopoietic stem cells, and MYC targets in the erythroid progenitors. But one of the main takeaways is that these cells are really not distinguishable from the wild type cells based on gene expression data alone.
Good point. So thank you. If there are no more questions, well, thank you so much for your talk. It's incredible work and really highlights the collaborations going on across disciplines. So to close up, this seminar has been recorded and it will be available soon on YouTube and on our website, which is multimodalintegration.org. You can follow us on Twitter at segs_ICA or check our website for the latest announcements regarding upcoming talks and events. And with that, I'd like to thank everyone for joining, and we hope to see you at our next seminar soon. Have a wonderful day.
Thank you so much.